we've been developing and how life detection fits into that architecture. Make sure the slides are working here. Most of our work over the last 20 years has dealt with enabling ocean world exploration. Since life will most likely be where the liquid water is, the engineering problem sounds kind of simple. You land, get to the water, and explore. We know how to land. It's the parts that come after that that are going to be hard. So for those who are not versed in the lingo, cryobots are self-contained ice-penetrating robots. I'm going to talk about 3 significantly different approaches that we have taken over the last 15 years. But first, let's talk about power. If we use Europa as an example, and of course any of the other ones out there are very similar, you have 3 choices. You've got solar cells, batteries, or nuclear power. When you work the ice penetration physics, it quickly becomes apparent that only the last of these is going to work. So in 2016, we spent a good deal of effort looking at nuclear heat sources. We designed the smallest fission reactor in existence, at least that we're aware of. And then we looked at the possibility of using things like excess strontium-90 from power reactors as a radiothermal source, largely because it's available and it's relatively inexpensive. And finally, we looked at traditional plutonium-238 in the legacy type heating units that NASA uses. All of these are currently in play. The big problem here is that while it is not illegal to take nuclear power sources to Earth's polar regions, the resistance to doing so is pretty high. So we need to come up with a nuclear surrogate to test these cryobots on Earth. And one way to do this is to optically transfer power to the vehicle using an optical fiber and an industrial laser. This wouldn't have been possible more than 10 years ago. Things have changed dramatically in that area. We were the first to attempt this back in 2010, and to cut a long story short, it works. We used this concept in the Valkyrie vehicle. There were actually 2 phases. This is phase 2. The laser beam was dumped into a heat exchanger at the front of the vehicle, and meltwater was then passed through that heat exchanger and then over to jet pumps that then emulated the very successful Antarctic hot water drills, except that this one is what you would call a closed cycle hot water drill. We tested this on the Matanuska Glacier in Alaska in 2014 and 2015 and demonstrated that the concept works and matches the thermophysics calculations. Those were the big ones. We were within just a few % of what we had predicted. It also proves that cryobots can steer in ice to avoid obstacles. We actually had an ice penetrating radar that we tested along with this that could look ahead about 100 meters and see objects about 1 meter in cross section, and within that we could deviate about 7 degrees off vertical. In 2016, we discovered a rather unusual wavelength dependent anomaly in the absorption spectra of photons in ice and water separately. And on the basis of that discovery, we began something called the Archimedes project. The setup for this was similar in many ways to Valkyrie, except that instead of sending the photons to a beam dump for heat, we collimated the beam and then focused it ahead of the vehicle into the ice, some 20 vehicle diameters ahead of the nose. So for those of you who do not believe in lightsabers and that they're science fiction, here is a lab test done here in Texas in 2016 that perhaps might change your mind. We also set a cryobot speed record with this test of 22 meters per hour. The efficiency of that system, a direct laser probe, is about 50% better than what you can get for traditional hot water drills, which is the best in the business right now. To put that into perspective, using a 60 kilowatt laser, we could send a 15 centimeter diameter science payload to the bottom of the deepest ice in Antarctica in about 5 days. However, if you want to send big payloads in deep ice on Earth, you need a different approach. There are limitations to the laser optics and the size of the vehicle. In the Thor project, we designed a full-size cryobot to carry an existing 50 kilowatt nuclear reactor as its power source in design, and we came up with a substitute surrogate for that, as you'll see here in a second. So here is a computer mock-up on the left, and the business end of the actual vehicle is on the right. The vehicle is 37 centimeters in diameter. That happens to match the NASA Glenn kilopower reactor. Obviously, smaller versions can be done. We did the design on those. Unlike all previous penetrators of this class, we used AC power and dumped that into a three-phase liquid resistor that emulates the reactor. On the surface, we have a 50 kilowatt three-phase generator driving a transformer stack to jump the power up to 10 kilovolts. And then down here, this is how we converted it. It's a liquid resistor, uses a titrated sodium carbonate solution in there to dump the power, and we were able to hit a power density of 600 kilowatts per liter, which is about 2 orders of magnitude higher than you can do if you're using traditional resistive heating. So here's a predictive performance plot for work on the Ross Ice Shelf using published temperature profiles through the ice. Using Thor and a large diesel generator, we can be through the deepest ice in the RS in about a day. Okay, so let's shift gears slightly. When we talk about a sub-ice ocean world mission, we're really dealing with 6 different regimes. The first 2 of these, the starting problem and cold ice cruise, can't be done naturally here on Earth. You need a simulator. So we built a special cryobot testing chamber. It holds a column of ice at 100 Kelvin and 1 millitorr, vacuum even under full cryobot heat load. And here is a quick movie. Let me see if I can get it going here. Showing a direct laser probe entering 100 Kelvin ice inside a chamber under vacuum. There's no melting at the start. The ice directly sublimates until the hole is closed behind the vehicle. Let's see if I can get this to go down here. Okay, within this context, there is a growing body of work looking at both how one should look for life, exemplified by the ladder of life detection, as well as what instruments could be carried by a robot that would shed light on those criteria in situ. We have so far had varying degrees of success with what might be called triage instruments, like the deep ultraviolet flow spectrometer that was carried on the Valkyrie cryobot in 2015. You can see it over here on the right. The cutaway is in the middle there. And we used that to trigger sample collection, autonomous sample collection that would then be brought to the surface for ground truthing by biologists who were doing parallel coring samples to see if the robot made the right decisions. Let me now jump to the other half of this equation. Deploying an AUV from a cryobot into a subglacial lake or ocean, we have built 3 of these subsurface devices for NASA, all of which, in addition to life detection and sampling objectives, also had to solve problems like navigation, guidance, control where there is no GPS, and perform other maneuvers like rendezvous and docking in the situation where the vehicle goes through a drilled access hole. In the DepthX project, we looked at using chemoclines in the world's deepest hydrothermal spring as triggers for machine vision-based behaviors to look for microbial mats living off hydrogen sulfide plumes. That simple behavior, about 30,000 lines of code, led to the robot collecting 4 new phyla of bacteria when fewer than 100 were known on Earth. Endurance was built for 3D chemistry mapping of subglacial Lake Bonny and the Antarctic dry valleys. Many people listening to this probably have heard of Blood Falls. Endurance discovered a completely new hypersaline anoxic water source at the grounding zone of Taylor Glacier without human guidance. Artemis here, shown being lowered through a 12-meter deep drilled shaft, was the first long-range AUV to do exploration under the McMurdo ice shelf. It had a complete suite of oceanographic and chemistry sensors, flow cytometers, and the like. But the most impressive thing it did was to return to a 1.2 meter diameter hole in the ice from 5 kilometers away and rise up the hole on its own. Hopefully we get time here for a couple more slides. Here's a very short video. This is mission control. We can talk about all this later, but here is the vehicle coming back from 5 kilometers out. It discovers an optical oscillating target source, hones in on that, latches it, and then signals to the surface to pull the system up, goes vertical, and is pulled back. The backup vehicle for Artemis was called Sunfish, which we developed independently here. It's person portable, and it's headed in the right direction to be cryobot deployable. The main advance made by Sunfish over the last 3 years has been fully autonomous behavior-based exploration in overhead environments. This video shows how we implemented real-time 3D SLAM for both navigation and exploration behaviors. Sunfish is our first commercial AUV. It's now seeing regular use for automated inspection and mapping. And so that was my last formal slide. To wrap it up here, just a few key points. The most important one is that cryobots and sub-ice AUVs are no longer science fiction. They're real, they're prototypes, of course, but they're becoming much more reliable every year. In fact, all the systems I've shown here are TRL 5+ on the NASA technology development scale. Next big step is going to be to conduct long duration analog testing in deep ice, which means kilometers deep. And the best place to do that is going to be Antarctica. From a terrestrial science perspective, cryobots are very likely to soon replace mechanical drills and hot water drills for access to subglacial lakes. And last, if you're out there and listening, we're looking for partners to put together proposals to explore deep subglacial lakes. Last slide here is more in the way of a public service announcement. Those of you who keep track in NASA research cycles know that we are now in the 2023 decadal survey, where ideas for planetary missions are put forth by the community. This time there's a very strong effort to prioritize ocean world exploration. And it's a subset of that. We need to be able to fully test the technology in analog environments. We're proposing a significant Antarctic field station, could be international in nature, for long duration testing of cryobots and sub-ice AUVs. You can read the paper at the first URL on this slide, and if you want to support the effort, please go to the second URL and add your name to the list. Thank you very much.
Absolutely amazing presentation, Bill. I think we're all just a bit like blown away by what's going on Earth. Thank you so much. There are lots of comments in the chat just about how amazed people were. So I'll just field a couple of the questions, and I'm sure this discussion will continue if you have some time.
Sure. Yeah.
So the first question is from Albert Fahrenbach. Is it possible to communicate through miles of ice with radio?
Yeah, so if you're posing that question, you may be aware of the, I guess, 23-year-old idea of sending out radio frequency pucks behind the vehicle. There are people who are looking at that right now. There are issues. There's always issues because of the unknowns, but we have actually tracked underwater view, the AUVs that we have. We didn't have time to show this, but we actually tracked that vehicle under the Ross Ice Shelf for quite a ways, not using radio frequency, rather using long wavelength magnetic oscillations down in the 3.5 kilohertz regime. We've been able to penetrate through 50 meters of seawater and about 100 meters of ice and track the vehicle to within about 1/4 of a meter of location. RF works if it's clean ice with no dielectrics. So that's probably a longer answer than you wanted to hear. If it's something of interest, contact me offline and we can have an hour-long chat on this.
Amazing, thank you. And there are so many more questions. Sorry we didn't get around to them, but yeah, if you could check it out in chat, I think everyone would really appreciate it. Thank you so much, Bill.
Be happy to, my pleasure.
Thank you. So next up, we have Chenoa. So might just ask Bill if you could stop sharing your screen, then we can switch over to the next speaker.
Right. Let's see here. Screen sharing has stopped. Okay, did that do it?
Yeah, that's it. That's great.
Okay.
Thank you. So now we're moving from exploration of Earth into looking outwards towards space. So Chenoa Tremblay from the CSIRO is now going to present to us on SETI searches at low radio frequencies. Thank you.
Okay, thank you. Sorry, it's still muted myself. Yeah, so I'd first like to thank the organizers for allowing me to share some of my research today. I'm going to go in another completely different direction. My name is Chenoa Tremblay. I work with CSIRO Astronomy and Space Science, and I'd like to acknowledge the Whadjuk people of the Noongar Nation as the traditional owners of the land I'm meeting on today, and the Wadri people as the traditional owners of the observatory site in which I use. And I would pay my respects to their elders past, present, and emerging. So I specialize in commissioning radio telescopes and try to find the unknowns and unknowns in radio astronomy data. Now, this is a complex challenge, and it doesn't offer a lot of interest to a lot of people, but I do have a lot of research in various different topics, and this will be the one I'll talk about today. In some of the search that we're doing in various different parameters. As we operate our telescopes remotely, we rarely get to go to actually see them. So as you can see in this photo, when we do get a chance to be there, we get to be tourists as well. So in addition to all of the wonderful science that has been discussed over the last few days of understanding life and its origins within our solar systems, including Earth, we have come to understand that expanding our search to the far reaches of the galaxy is also important. Despite the science fiction books and movies like to tell us, unfortunately, we still can't travel faster than light or even as fast as light. So we use telescopes to search for light that has produced small discrete frequencies across the electromagnetic spectrum. Since the late 1950s, a lot of research has gone into searching using radio wavelengths. As the technology is relatively cheap in comparison to some of the others, radio waves are not stopped by the gas and dust within our galaxy, and there are a large number of photons or packets of light. Most of these searches are done using telescopes that look something like this. Big concave radio dishes that often cover wavelengths of centimeters to millimeters in size. Although these searches are important, and since we don't know when, how, or what a signal may actually look like, searches of different wavelengths and of many different telescopes are really important. With my research, we're trying something slightly different. We're using these spider-like dipole antennas. Each are a grid of 16 independent antennas that work together as a tile. And there are 256 of these tiles spread over 5.5 kilometers. This is called the Murchison Widefield array, and it is searching in meter wavelengths, similar to what car radios and digital TVs produce. For those that might be interested, the telescope observes in the frequency range of 70 to 300 megahertz, a bandwidth of 30.27 megahertz, for a single observation, and a frequency resolution of 10 kilohertz. This telescope, as well as the Australian Square Kilometre Array Pathfinder, are precursor telescopes to the site of the future low frequency Square Kilometre Array, situated in the outback of Western Australia. Now, this site is ideal for this type of science as it's hundreds of kilometers from any city, and thus reducing the chance of terrestrial interference in our astronomical data, which is quite sensitive. Now, radio telescopes, including the MWA, can be used in a number of different modes, depending on the science people are interested in. I turn the telescope into a spectrometer, and spectroscopy is the study of the interaction of light and matter. We can break up the wavelengths of light and detect the astrophysical objects into very fine detail and plot the signal strength as a function of frequency towards a given object. This spectra is similar to what may be produced by an ICP-OES or an ICP-MS that has been discussed over the last few days. The difference here is I don't have control over the plasma that's generating these emissions. So from here, we can study the phenomenon like emitting molecules and other types of objects in space in various different ways. Other than a different observing wavelength compared to other telescopes, the MWA also allows us to view a very large portion of the sky all at once. It views up to 900 square degrees, but due to processing constraints with the very large volume of data, our images tend to cover between 200 and 600 square degrees. Now, for perspective, this is roughly equivalent to taking the area of the sky, taking 1,600 moons, laying them out side by side into a square. So a very large volume of space. The region most recently studied was toward the Vela constellation, as viewed in this image. Here, there are a number of supernova remnants, which are kind of the bubbles that you see, or the flower-like structure in the center, caused by exploding stars, ionized gas regions where new stars are forming, and thousands of galaxies and millions of stars, all in the single image. In my work, I use astronomical data to look for signals that change as a function of wavelength, to look for changes associated with sharp electronic signaling or technosignatures and molecules as tracers for life. By looking for gas phase molecules, we hope to detect life that may not have developed the intelligence to send out its own signal, or help understand where the seed molecules for life may have come from. In radio astronomy, we are searching for rotational transitions or emission of molecules that may change speed. So I'm going to start by talking about these electronic signaling. To determine a metric for how we would expect to see a signal if one was purposely sent to us, we use an experiment that was done by Arecibo collaboration in 1975, where they sent a purposeful signal at a high power to determine the strength as a function of a distance that we may expect. This transmission, which we call the equivalent isotropic transmission power, of 20 times 10 to the 9th watts, and represents a model for what we'd like to achieve for sensitivities for signals from nearby systems. In our recent paper that was published this week, we were able to calculate this equivalent isotropic radio power for over 10 million stars, thanks to the Gaia survey that provides the distances. Now, it was suggested that approximately 20% of galactic sunlight stars could have Earth-sized planets in their habitable zone, and Kipping in 2020 paper suggests that the search for technosignatures from stars with stellar types much earlier than our own sun may not be necessary, as life is unlikely to ever evolve. And this is where Gaia becomes really important, because its sensitivity is perfectly centered in this range. Now, in this paper, we didn't detect any unknown astronomical signals, but what we did find is that we still have about 3 orders of magnitude in signal strength to go before we can start actually expecting to even possibly detecting something. But there's a lot of other complexities to that as well. When we look at our work in comparison to the landscape of other searches that are being done by radio telescopes, we are exceeding the limits of some of the other surveys by an order of magnitude due to our wide field of view, being able to see so many stars at once. Now, in other work submitted for peer review, we're looking for simple molecules that are interesting for their biomolecular tracers or thought to be components of more complex amino acids or other molecules. At least for what I understand, it would be interesting to know if there's any other people at this conference today that have other ideas on what other things we should be looking for. As a space is low density, it is expected that a good fraction of the molecular interactions are 2-body interactions, as opposed to the density of Earth, where 3 or more interactions may happen to form more complex molecules. We have completed new molecular modeling to determine where the emission of nitric oxide in particular would be emitted between 8 and 4,000 megahertz. We are combining this information with searches for hydroxyl and methyldiene, CH, in both emission and absorption to study the early stages of star formation. Absorption can be seen as we look towards very bright objects in our images. To go with the new molecular modeling, we also completed a blind spectral line survey of 17 hours of data using the MWA, down to a limit of 0.21 Janskys in intensity. We did not detect any molecules in emission or in absorption, as shown in the spectrum on the left. The right shows the RMS of the data cube. So if we calculate that along the spectral axes, we can get this to understand our noise properties and the Gaussianity of our noise. We are finalizing the referee comments on this paper, and we hope for it to be accepted soon, as it's been well received by both reviewers. All of this work is leading to understanding the challenges that we are going to face with the future square kilometer array. It is expected the telescope will be 50 times more sensitive than that of the MWY, but it also means that the data challenges are going to be even greater. But the science should be quite exciting, and I'm looking forward to it. If any of this is interesting of you, I'd love to chat and diversify my collaboration that is already made up of astronomers, theoretical chemists, theoretical physicists, and engineers. Thank you very much, and I'll take any questions.
Thank you so much, Chenoa, for that fantastic talk. So I'll start by asking a question myself. So when you show that you're using the WMA as a spectrometer, is the spectral resolution limiting at all, or is it adequate for what you want to study?
Yeah, so the current spectral resolution of the telescope is quite broad, and it would be better if we could narrow that spectral resolution, and that will help with both a confusion if we could identify a feature and what it actually represents, for which molecule it might actually represent, and being able to understand the motions of the objects in which we're detecting them, being able to see the shift in frequency to interpret that as a velocity of the objects and the motions. So there is an expectation that there will be an upgrade to the telescope over the next year. They're working on getting funding at the moment, but they've already trialed the systems. So that would be, I think, a factor of 100 better than the current frequency resolution.
Oh, amazing. I guess for us bench scientists, we kind of use little UV vis machines or infrared spectrometers, but just have no idea how for an array of instruments, like what is the modification that you make to get that boost in resolution?
So in particular, this one, as you can kind of see on the screen, it's made up of all these different tiles, and we combine those together to mimic the strength of a single essentially big antenna. And when we do that, we have to combine those signals together into a process called correlation. And so really the difference is in processing power. So to be able to get better resolution, we need to improve our technology, our supercomputers, and our software, and to some degree, some of the electronics that are sitting on the ground, like the box I'm standing next to in the picture, in order to get that better resolution. But a lot of it comes down to software.
Yeah, that's actually really intriguing. I don't think software is like usually an issue in the small scale bench science that we do. So
Exactly.
Thanks for that insight. I guess what would be the first question that you'd want to answer once you do get those upgrades?
Well, one of the things is that no one's ever been able to detect molecular transitions down at these lower frequencies. So the last, I guess, 5 or 6 years, I've been trying to develop all the software necessary to try to do this experiment, and then doing all the preliminary understanding for what we would expect to detect, how we could do it, and what other changes do we need to make, and what other understanding do we need to understand from a molecular standpoint, a theoretical standpoint, and things like that. So being able to prove that, it is not only theoretically possible, but it is physically possible to just start detecting molecular transitions down at these frequencies would be wonderful. There's a paper that came out, I think in 2012 or 2013, that suggested that if we wanted to detect glycine down at these lower frequencies might be the better way to go. But in order to do that, you need something like 1,000 hours of observations. As a standing, or I guess as a comparison, 17 hours of data was hundreds of terabytes and over 400,000 hours of supercomputer time. And so to process that onto your laptop for just 17 hours of data would have taken me 25 years. So trying to process 1,000 hours of data, it seems like an enormous challenge at the moment, and something that we kind of have to work out still.
Wow, that's incredible. Yeah, buy more servers, more computers and processors. But yeah, that's fantastic. And apparently your paper's making the rounds on the internet. Graham Lau from the Blue Marble says that your paper's semi-famous. So thank you so much for sharing your work with us. And yeah, it's very exciting stuff.
You're welcome. Thank you.
Okay. Next up, still in the theme of space, we have Nicholas Rattenbury back for a talk on a completely different topic from 2 days ago. So would you like to share your screen?
Yep, there we go. Can everybody see that?
Yep, fantastic. Okay, thank you, Nicholas. I'll let you take it away.
Alrighty. Ngā mihi nui ki koutou katoa. Ngā mihi nui ki te tangata whenua. My greetings to all of you, and my greetings to the tangata whenua, the people of the land. Yes, this is the second talk in this conference from me, so thank you very much to the organisers for obliging me. It is a completely different topic. I'm talking about the space systems research that we are conducting at the University of Auckland. This is work that has a very, very short history. 5 years ago, I was solely working on the topic of my first talk, which was the discovery of extrasolar planets through gravitational microlensing. And increasingly, over the last few years, I have pivoted towards supporting the research and the vast amounts of administration which goes into setting up a brand new program for undergraduate and postgraduate students in designing and building space missions. So I'd like to talk to you a little bit about how that is going along, if my computer will behave. All right. So I'm an astronomer, despite the fact that I have started to move towards space systems engineering. I'm a physics lecturer in the Department of Physics at the University of Auckland. And I'm also a member of Te Pūnaha Ātea, the Auckland Space Institute. This is a new institute which has welcomed recently, in the last 10 months, our first director, Guglielmo Aglietti, that comes to us from Surrey. And with us, so with him, we have Jim Hefty on the right there, and Tristan Hanlon, who take care of the undergraduate program for space systems, the APSS. That program is a not for credit, it is a purely voluntary program for undergraduate students from across the university to design and build a space mission of benefit to New Zealand, which could fit in a 1U CubeSat. A CubeSat, 1U CubeSat is these really tiny little micro satellites, 10 centimeters to the side. At the time when we first started off, we did this with initial seed funding from the university internally of $150,000. And if anybody knows anything about space systems, you'll realize that that doesn't buy you an awful lot of satellite. In fact, it doesn't buy you, well, even one satellite. However, thanks to the work done by our undergraduate students in the undergraduate program, we have attracted funding for the next 4 satellites from a private philanthropic source, which is fantastic. We're able to fly the next 4 satellites in that program. And partly on the strength of that and the interest of the researchers at the University of Auckland, and also naturally the interest in space systems, with the advent of Rocket Lab USA starting to launch rockets from New Zealand, the university decided that they would dedicate a great deal of time and resource to attracting staff to come and build up the Space Institute. And this is how we've come to where we are now. We are welcoming more and more staff from across the world to work with us, design and build our research program. The interim director before Guglielmo showed up was Professor Richard Easter, a name and a face you'll see shortly more of in a second. We are ably helped by Catherine Poltro, and if you're on Twitter and you follow anything to do with astronomy, then you can check out Kathy Hugh or Catherine Hugh's Twitter feed. John Cater is my counterpart in engineering. He and I work together to get students through their research projects in the undergraduate and postgraduate topics that they're studying. We have another research institute at the University of Auckland, and that is Te Awamarama, the Centre for Fundamental Inquiry. And we start off with, well, trying to tackle these big questions. What is life, for instance, and how did it start on Earth? Did it take hold elsewhere? And the Centre for Fundamental Inquiry, Te Awamarama, has a number of high-powered people in it, including one of our contributors to this meeting, Kathy Campbell, and again, there's Richard Easter, physics, and a whole bunch of other people, including myself and John. But we include people, biologists, theoretical biologists, philosophers, Te Ao Māori specialists, computer scientists, and environmental scientists, all with the same goal, which is to answer these massive questions like, how do you define even what life is, much less how do you go about find it. and we work in this research institution with the view that there's people across the university in all walks of research background that can help us tackle this problem, or even start to refine what that question actually means so that we can answer it in a sensible way. For instance, you can take a look at hot springs as a source of life, the warm pond theory that is compelling for the origin of life on Earth. How does that tie in with space systems? Well, let me tell you a bit of a story. This is a little bit of a background on some of the major research themes that we are pursuing at the University of Auckland. One of them is looking at novel ways of pushing a satellite around the solar system. And we're looking at the sort of propulsion systems which rely not on chemical reactive rockets, but on throwing charged ions out the back of the spacecraft to push it further forward. And we have a couple of PhD students, Felician and Antonella, who are working with us, and also Christine Charles and Rod Boswell at ANU in the Space Physics and Plasma laboratory there, looking at designing novel propulsion systems for these little satellites. Our work is focusing largely now on ionized plasma propulsion, particularly magnetohydrodynamic physics within these charged plasmas. There is an image of an experiment at ANU which we are trying to replicate in Auckland. The start of our lab bench, which is very nice. We've got lovely vacuum chambers from designed from ANU, working on, in this case, an experiment to run this little pocket rocket unit here from ANU and Stanford. And Antonella and Feliciana are starting to work on building a much larger unit here, which has interesting physics, which is not fully explained. Some breaking news fresh off the press. As of 1 hour ago, just been informed that a research group which we're working with has been awarded $11 million from the Ministry of Business, Innovation and Employment to look at funding research into high magnetic field electrical propulsion for space. So it turns out when you need to generate these sorts of plasmas and take a look at their propulsive properties, you need to generate really strong magnetic fields. How you do that in space is a bit of an open question, but that is what this research is being funded to do. The PI is Nick Long at the Robinson Research Institute in Wellington, and on that grant is both myself and John, and we are looking at the space systems subsystem platform development that is going to enable the sort of experimentation in space. I need to point out that there's a whole bunch of people on this funding application, including the colleagues at the University of Canterbury and a whole host of others. So this is exciting news for us. We have suddenly got a whole bunch more work to do in this field. If you've got a rocket or you've got a propulsion system, you'd like to know which way to point it so it gets its way through the solar system for exploration purposes. And the common feature, if you like, of those plasma propulsion systems, as opposed to a chemical rocket, is that they have very low specific thrusts. They don't push very hard, but they push very efficiently for very long times. So all of the mathematics which has been developed over the last 30 or 40 years about how to push a spacecraft around in the solar system is predicated on the fact you've got a great big fucking chemical rocket out the back which can push you pretty hard, pretty fast anywhere you want to. The mathematics and the computing algorithms which embody those mathematics have to be challenged now, because instead of having a really powerful chemical rocket which has different characteristics to these plasma propulsion technologies, it has to be challenged and the mathematics reformulated. So work which is being done at Auckland, led by Darcy Graham, a PhD student of ours, is looking at ways of changing the mathematics and embodying those in a way that you can optimize these spacecraft trajectories, assuming you have a very low delta V thruster on the end. And so she's been working on this sort of work. There's a diagram here to simplify it. How do you get to from Earth to Venus, for instance, using a spacecraft being pushed around by one of these very low specific thrusters? And she's been working on that and recently published or presented her work at the American Astronautical Society. I can probably share a link to this presentation. There is a YouTube video of Darcy giving the presentation, because it was all about the COVID and we had to do this remotely, so she sent in a nice video. We're also looking at novel material science, in particular looking at making a foam out of titanium. New Zealand has a local titanium industry. We have a lot of titanium around, and we have some good local research being conducted on the behavior and the qualities of titanium, in particular titanium foam. So we're looking at turning titanium into a foam to become a novel form of heat shielding, very, very light, and a lot of attractive properties for this work. So Philip Necker is another PhD student working with us. He is developing these titanium foams, and the idea is to take them and put them into a plasma wind chamber and see or watch them as they dissolve and get vaporized, and measure their qualities and their usefulness as heat shielding material. And this is research funded by also with down at the University of Ātea is a fantastic researcher who's doing work under a National Science Challenge and a Catalyst Fund to investigate how you can conduct biological experiments in space, leading to a more understanding of life on Earth. So understanding how biological processes occur in space helps us understand how they indeed worked on Earth, plus also elsewhere in the solar system. The work is on developing recoverable laboratories. The idea being is that you take a sample of reagents in space, experiment, you need a small light to do that, possibly an experiment from the ISS. But also if you're able to develop this sort of experiment which is light, easily transportable and recoverable, you can start to look at the properties of the materials which were created in orbit or through the field, or indeed making measurements looking for precursors of life elsewhere in the solar system. So this is coupled with Philip's work on titanium heat shielding. If you want to be able to protect your experiment going through an extreme environment, such as some heat, the heat of re-entry or atmospheric entry, then you have some way of protecting. You have a heat shielding material which will protect your laboratory experiment. This particular work, though, that Sarah is working on is a proof of concept looking at crystallizing proteins in low Earth orbit. And again, both John and I are working with Sarah to provide advice on the mission operations concept and the subsystems of the satellite that will be able to enable that. Of course, we can't have any sort of talk about this sort of work without a nod to Rocket Lab USA, which is a company in New Zealand, which has recently announced their interest in getting a spacecraft to the moon and indeed further afield. This is an image of their Photon kick stage, which has been adapted, it's an artist's rendering, has been adapted for basically interplanetary flight. And we want to leverage off this new interest and capability to further our research in this area. To that end, we are interested in scoping out a mission to Venus's atmosphere. Venus's atmosphere is a possible avenue, a possible region, a possible volume wherein we might find something interesting about the emergence of simple life. So the problem we start off with is, what is life? How do we find it? And that is something which we are trying to tackle with extended subject matter expertise within Te Ao Marama, the Centre for Fundamental Inquiry. Working with people such as Sarah Kessan's University of Canterbury, who's developing an experiment, for instance, which will be able to discover precursors of life. Working within Te Ātea, the Auckland Space Institute, to design suitable missions to carry those experiments places which are of interest to us, including the atmosphere of Venus. So I hope I've tried to sketch out here. We're trying to put together all these ingredients for this sort of capability. We've got propulsion expertise being developed. We're looking at utilizing that propulsion in the smartest possible way with our trajectory optimization analysis. We're looking at exquisite lab experiments in situ using our heat shielding material science research, and we'd like to talk to launch providers such as Rocket Lab, who are looking for opportunities to give people like us a lift to our targets. So that is pretty much what I wanted to talk about. This is a quick summary of the work that we're doing, and apologies if I have gone over time.
Pretty spot on. Thank you so much for showing us all the amazing work happening over at the University of Auckland and collaborators. It's super impressive. I might advise everyone to go continue the conversation in chat. Thank you, Nicholas, for that great talk. And I'm going to ask our next speaker, Bonnie, to start sharing slides, please. Fantastic. So now we're switching gears. We're now looking at sort of the preservation of records on Earth. So thank you so much. We have Bronwyn Teece starting off this part of our afternoon with a talk on organic matter from Fossilised Hot Spring Sinter from Yellowstone. So thanks so much, Bonnie. Take it away.
Thank you, Anna. So hi, I'm Bonnie and I'm a PhD student at the Australian Centre for Astrobiology at UNSW, and I'm also an organic geochemist. So today I'm going to be talking to you about fossilised organic matter in hot spring sinters. But before I begin, I'd like to acknowledge the Aboriginal and Torres Strait Islander peoples, the first Australians, and specifically the Bidjigal people on whose lands UNSW stands, and the Gadigal people on whose lands my own house stands, of the Eora Nation, on whose lands, winds and waters we all share, and I'd like to pay respect to their unique values and their continuing and enduring cultures, which deepen and enrich the life of our nation and communities here in Australia. And for those of you who haven't seen this map, this is a map of the continent that we now call Australia, and it's made up of over 200 Indigenous countries. I would also like to acknowledge and pay my respects to the First Nations people in what is now known as the USA. There were 26 tribes who held ties to Yellowstone over 11,000 years. So I'd like to thank them for their custodianship of the land from which we benefit and learn from. So today I'm going to be talking a little bit about silica precipitation in hot springs. So there are also carbonate-based hot springs, which Kathy mentioned on Wednesday, travertines. But for the purposes of this talk, I'm focusing only on silica hot springs. And what happens in a silica hot spring is that there's a lot of hot water that's supersaturated in silica below the surface. A geyser will explode, bringing this supersaturated water up and out where it cools down and precipitates outwards on what's known as a silica sinter apron. So imagine you're a little bacteria and you're floating along, minding your own business, and then all of a sudden you die. Well, then you fall apart and your body's probably lost because you're soft-bodied. But not in silica sinters. You see, this precipitation is sort of like a bacterial Pompeii, entombing these microorganisms where they sit and showing exceptional preservation in these environments. We also can see this in the fossil record looking back. Most of the ancient microfossils are entombed in silica, although in chert. And we have microfossils preserved from 3 billion and 2.4 billion years ago, among others, that are exceptionally preserved because of the way that silica entombs around organisms. But there's a complementary story. When we think about the fossil record, we typically think of physical fossils, but there are also biomarkers or chemical fossils, that have a complementary story. So what happens is that when organisms die in this model in an ocean, they fall to the ground and they're concentrated within the sediment. As they're concentrated, they undergo diagenesis, which is a reaction of fossilization, which transforms this organic matter to more stable compounds. So a lot of talks in the past 3 days are focused on things like fatty acids or DNA, and these are the less stable compounds. So if you take this biolipid right here, hopenpyrrole, which is a biolipid found in the cell membranes of bacteria, once the bacteria dies and it's buried, a diagenetic process can transform this, taking away the OH groups to a more stable group called a biomarker. And this biomarker is hopane, which is also a biomarker for bacteria. So why is it important to understand organics in hot springs? Well, there's a variety of reasons. As you probably know, opal A silica has been detected on Mars at Columbia Hills. There's been a series of papers looking at the detection of these minerals and what that means, as well as one-to-one comparisons with hot springs like Ātea on Earth to show that this environment was most likely a fossilised hot spring. We also know that hot springs were prevalent on the early Earth in the Dresser Formation, and they may have been the site of the origin of life. So either way you slice it, understanding hot springs is important for understanding either early Earth or early Mars. But our research into organic preservation in hot springs is quite limited. Most of the studies focus on these polar compounds, the fatty acids and the biolipids, rather than the geomolecules that have been preserved in the record. And most of the studies also focused only on samples that are less than 1,000 years old. When you're thinking about 3.5 billion-year-old fossil environments, 1,000 years isn't much on that. So we really need to expand our knowledge of preservation of organics through the fossil record. So earlier this year, I had a paper with some colleagues that came out, and we looked at modern Sinters from Ātea and compared them with some fossilised Sinters from New Zealand. And what we found was key was it wasn't actually the age of the Sinter that dictated the likelihood of preservation of these hydrocarbon biomarkers, but it was actually the degree of diagenesis that had occurred in the Sinter. So this is really important when you're thinking about somewhere like Columbia Hills or Mars, where the silica is still pristine. Even if it's old, that doesn't matter so much. What matters is the crystallinity of the silica. So what about other fossilized hot springs? Well, there's not much information about that. So we decided to investigate some hot springs from Yellowstone National Park. And I'm just going to talk you through a few of our samples. So this is Steep Cone Sinter. There's no scale bar in this image, but the field of view is about 2 meters. And we took 3 samples that are quite close together from Steep Cone, and they're about 7,000 years old. We also took a sample that I separated into filaments and base, and this sample came from the rim of a hot spring, where this base became more silicified than the more friable filaments that were attached to it. There's also a sample that we affectionately refer to as Biffy, and this is a sample from an iron-rich silica hot spring. And I've just included here some shots of the thin sections so that you can see how much life there is. These are all filaments, and within Biffy, this beautiful sample, there are all these little pyres of stromatolites. It's just like Kathy said on Wednesday, in hot springs, life really is everywhere. So my colleague Jeff looked at the steep cone map textures under SEM, and what he found were bundles of filaments all lined up together. And near the bundles were gaps that had not been filled in within the past 7,000 years. You can actually see this botryoidal silica precipitating on the sides, but there's this huge gap. So it shows that there hasn't been much fluids pushing through, precipitating new generations of silica. In the sample that looked like filaments with the base, you can also see under SEM these networks of tangled filaments rather than bundles of filaments. And inside these tangled filaments, after the bacteria have disintegrated and gone away, they've actually filled with silica, replacing the internal texture. So I decided to look at the organic geochemistry of these samples to see what we could find. So I'm just going to walk you through this graph. The bottom here is the straight chain carbon number, and the top side here is the nanograms per gram of organics that were in the sample. So one thing that's key here is that the 3 samples from Steep Cone, although they were so close together, have really, really different signatures. And that shows an incredible diversity over a very small scale. We also see that in Biffy, the iron-rich sample, there was a high abundance of organics relative to the other samples, which is quite intriguing given that iron doesn't usually enhance the preservation of organics. For the filaments and base, you can see this really smooth distribution, which contrasts sharply with these jagged distributions in the other samples. And this is unusual in recent sediments because a smooth distribution really usually means that the organic matter has been matured, which would not be the case here where the sediments haven't buried, unless the organic matter is being brought in from elsewhere, maybe deeper in the hot spring system. The last thing that I want to draw your attention to on this graph is this C17 straight chain hydrocarbon. So an enrichment of C17 relative to the neighboring alkanes is a biomarker for cyanobacteria. This is well recognized within the organic geochemical community. And what's actually even more exciting is that when you look in between C17 and C18, you can see a distribution of branched analkanes that can tell you about the bacterial communities within the rock that you're looking at. So this is a sample of a Sinococcus chloroflexi mat that was taken by Nikki Parental et al. In 2014 from chocolate pots. And they took this mat and they ran it through a GCMS. And this is C17, this is C18, and these are branched analkanes. And so what happens is that with different bacterial groups, the relative amounts of these peaks to one another changes. So what I was actually able to find was that in 2 of my samples, we could see signals of these Sinococcus and chloroflexi mats. This matches with the metagenomics of this area. And this is really exciting because DNA does not preserve through the vastness of geologic time, but these hydrocarbon biomarkers do. So if we have a good understanding about these in more recent sediments, we can start to look at them in deeper time and begin to reconstruct the communities that grew in these environments. So in the next slide, I'm going to show you a series of pie charts, and they're going to just show you the different distributions of these peaks to one another in different samples. So if you're not an organic geochemist, just ignore this key. It's not relevant. I just left it there for anyone that might be interested. The key takeaway that I want you to see here is the diversity within these samples. So these 3 samples that came from Steep Cone, they were so close to one another that you can see that the bacterial communities are completely different. For the base and filamentous sample, you can see that the communities are quite similar, as you would expect. And for the banded iron sample, you can see that it's different again. So this shows a huge diversity within the communities of bacteria that we can see just among these few pools. In our research in Ātea and in New Zealand, we also found different distributions of these compounds, which show a means of tracking the different bacterial communities deep into the fossil record when metagenomics are not available. So the key takeaways are that in these hot springs, there's a huge diversity, even on the small scale. This is really important when you think about sampling, especially in hot springs on Earth, but also elsewhere. We really need to track down where these organics are sitting and where these microfossils are sitting within the samples. Another key takeaway is that these are well preserved, not only the physical fossil, but the organic molecules as well. There wasn't a large difference in abundance between the modern mats and what we were seeing from the fossilized molecules. So that's really interesting. But there's also a need for more geological studies because we've got all the rest of this breadth of geologic time that we don't have any data points for. So we really need to extend this back. And this is really important for looking for life on Mars because, of course, the more we understand the preservations of organics in hot spring centers, the more we understand whether Columbia Hills is an attractive target to return back to look for organics. Thank you.
Great, thank you, Bonnie. Fantastic talk and really exciting for me as a lipids person. So just have time for a quick question. So this is from Brendan Burns. So great talk. And also, are you seeing archaeal hydrocarbon signatures in your samples?
No, and it's quite surprising, actually, not just archaeal signatures, but in Ātea, the Sinter is actually constructed almost entirely of diatoms. It's quite diatomaceous, and I don't see any diatom biomarkers in the silica Sinter either. So it may be that there's a preferential interaction with the certain structures of molecules and the Sinter, but I'm not quite sure what's going on there and why only some groups are preserved better than others.
Okay, fantastic. Well, thank you so much, and might get us to switch over for the 2-part talk coming up next. So thanks, Bonnie. So next up, we have a special 2-part talk by Dr. Erica Barlow and soon to be Dr. Georgia Soares. Yes. So this is continuing on the theme that Bronnie has brought us into. So I don't know who's going first, but would one of you like to share your screens?
Yep. So can you hear me?
Yes, I can hear you, Georgia, and your screen looks great. Thank you.
All right, sweet. How do I get my video up? Oh, probably doesn't matter. All right, good afternoon, everyone. I'm Georgia, and today Erica and I will be talking about environmental change driving biological complexity in the fossil record. So let's start off briefly with the fossil record. So about 3.5 billion years ago, we have a first appearance of life in the form of stromatolites. At about 1.65 billion years ago, our ancestors, eukaryotes, have appeared. And at about 600 million years ago, we have the Ediacaran fauna and the rise of animals. At the same time, you have great environmental changes with two big rises in oxygen, the Great Oxidation Event at around 2.4 billion years, and the Neoproterozoic Oxidation Event at about 600, 700 million years. Accompanying these are periods of glaciations, and following these glacial periods are an influx in nutrients. And when you combine these two records together, you can see that in the Neoproterozoic, with great environmental change, an influx in nutrients, oxygen, and following periods of glaciation, you have the rise of more complex life in the form of the Ediacaran fauna and animals. Eukaryotes appear in the boring billion, although it has been suggested that they actually evolved earlier, but we're not exactly sure when because of the poor fossil record. Because of the event in the Neoproterozoic, it has been suggested that the great environmental change during the Great Oxidation Event led to the development of more complex biology. However, because of the lack of fossils from this time period, it's previously been really hard to test this theory. Until now, we have a 2.4 billion-year-old microbial reef complex within the Turi Creek Group in Western Australia. This reef complex is diverse and abundant and has lots of stromatolites, microbialites, and microfossils. And this gives us an insight into what life was like in this time period. This reef complex spans from shallow to deep waters, and it contains two new novel fossil forms that don't seem to have any analogues in the fossil record. The first being what I'm studying, sedentary macroscopic life from shallow waters, and the second being what Erica discovered, planktonic microscopic life preserved in deep water cherts. And Erica will be talking about this in the second part of the talk. For now, let's see what I found. So these structures are attached to stromatolites. They're branching. They're made up of organic matter, which is the darker sections, and silica, which is the lighter sections. And so they're termed branching organic siliceous structures, or BOS. They grow upwards and outwards from stromatolites and have clear attachment points to the stromatolites, anchoring them onto stromatolites. They also have a really interesting morphology. So they start out as these bud type BOS with these little knobs growing off them. These knobs then grow into cylinders and simple branching structures, which still have quite a bulbous base reminiscent of the bud type origin. From these initial branches, secondary branches grow, forming these multi-level branching structures. And these multi-level branching structures can grow up to 3 centimeters tall. They have multiple branches growing from single points along specific points of the BOS's structure. Interestingly, these more mature forms have flatter to more triangular bases. And so what you can see is that these structures have a distinct and complex morphology. BOS are also within intercolumn sediment that contains phosphatic pelloids made up of carbonate fluorapatite, and within BOS themselves, associated with the organic matter, the carbon here, is apatite. Sedimentary apatite is evidence of an influx in nutrients in the system and also potentially oxygen. To test this, we did some trace element analysis, and we got a cerium anomaly on average of about 0.7, but it can also be much smaller. And so these negative cerium anomalies are indicative of oxygen. And so BOS are growing in a nutrient-rich, oxygenated environment. This environmental context combined with the morphology can narrow down what type of life BOS possibly represented. While boss were going 2.4 billion years ago, microbialites were the dominant form of life, and so we'll start there. Brutaxites are a type of microbialite with a simple branching structure similar to boss. However, their chemistry is really different. They're made up of manganese and iron, and they are far smaller than boss, and this rules them out as a candidate. Also, there are microdigitate stromatolites, and they have a similar simple branching morphology. However, these branches tend to be strictly parallel to each other. Also, microdigitate stromatolites tend to grow, the branches tend to grow into each other and out of each other, and their morphology is dependent on sedimentation and aren't an inherent characteristic like with boss. So we have to step up a level of complexity. So there are epiphyton, which are prokaryotic cyanophytes, which have inherent characteristics. These segments form together to make bigger branching structures. And when you zoom out, you can see that these structures are actually quite bushy in nature. They grow together and form a boundstone fabric, which is unlike that of boss, which form individual structures. And they're also much smaller than boss. So this partially rules them out. The best comparative morphology is with eukaryotic algae, green algae. For example, they have specific branching from specific points along the structure. You can have multiple branches growing from a single point, and they have attachment structures similar to that as boss. So you can see that boss resemble quite a range of different types of life. However, they have a better comparison to the more complex life. And so you can see that regardless of whether they're actually prokaryotic or eukaryotic, they are a step up in complexity compared to contemporary microbialites. And so you have this more complex organism living in a shallow water, nutrient-rich, oxygenated environment following glacial period. And now Erica will take you through what she's learnt throughout her PhD.
Thanks, Georgia. So I discovered these particular microfossils during my honours project in the Pilbara of Western Australia back in 2013. I had no idea what they were, and I couldn't find anything from the fossil record that looked like them. So as part of my PhD, I documented these fossils in detail to try and determine what kind of life they might have been. And I'll share some of these details with you now. So these microfossils are contained within nodules of black chert, shown here in rock outcrop. The chert is made up of very, very fine quartz crystals, which have the dual effect of encapsulating fossils and organic matter while minimizing alteration of this material by fluids circulating during diagenesis and metamorphism. So black chert is in effect like a time capsule, allowing us to see what life was like billions of years ago. So this is a thin section image with the microfossils highlighted by the stars. All of the dark brown material in this image is organic matter, so you can see just how much material can be preserved within chert, given the right conditions. Zooming in, these microfossils are spherical in 3 dimensions and are as large as 143 microns in diameter, so they've been descriptively termed the large spheroidal microfossils. They have this really distinctive clear spherical rind that when you look under the microscope in cross-polarized light, you can see is made of finer crystals of quartz than the surrounding material. This shows that the clear spherical rind was not an artifact of a silica mineralization front having pushed organic matter away from the main microfossil body, but rather that the clear spherical rind was likely an original feature of this microorganism. Looking internally, the large spheroidal microfossils contain a honeycomb-like texture, which is quite clear when you view through a confocal laser scanning microscope. In this view, the honeycomb-like texture actually forms more of a framework that is mostly circular, but in some places slightly polygonal. In really well-preserved microfossils, there is still this coarse honeycomb texture, but also what appear to be cell-like spheres. In another well-preserved example, you can see these really tiny, tiny cell-like spheres. There's more on that later. Another distinctive feature of this microfossil are these thin, irregular, wire-like features that extend out from the main microfossil body into the surrounding rind. And just for clarity, I've annotated here what I can see under the microscope in thin section. Just a bit hard to pull it out on the slide there. So as I mentioned, these microfossils, there's no known counterpart in the geological record that I've been able to find. The only analogous form that I could find were modern synovial algae in the Volvaceae family. As you can see, there are some striking similarities in overall size. All of the images here are at roughly the same scale, but also the spherical nature and the presence of a clear spherical rind, which in the case of the extant algae is a mucilaginous membrane that helps to keep the cells of the colony together and the environment out. There are also other similarities, including thin wire-like structures. In the case of the extant algal cynobia, these structures are called flagella, and they help to move the colony about in the water column. In addition to the mucilaginous membrane that surrounds the extant algal colony, each of the cells within a colony also have a membrane. In some examples, these cellular membranes are connected, forming a framework structure with circular compartments. So perhaps the presence of extracellular membranes could explain the honeycomb-like framework in the microfossils. In other examples, the tightly packed nature of cells within an algal colony creates polygonal, almost hexagonal boundaries. The Turi Creek group fossil displays a similar feature. And when the extant algal colony reproduces, each cell divides into 16 or 32 daughter cells, depending on genus. And this can happen asynchronously, which means that some cells are fully divided, while at the same time, other cells haven't even started dividing yet. So perhaps asynchronous cellular division could explain the textures that we see in the microfossils. Overall, the level of morphological complexity displayed by these large spheroidal microfossils, combined with the suite of morphological features that are analogous with modern synovial algae, suggests that these microfossils were more likely to have been eukaryotic rather than prokaryotic microorganisms. In either case, these microfossils display a dramatic increase in complexity compared to other Archean and Paleoproterozoic microfossils. So, putting all of this together, what is the significance of these fossils? Well, recall that Georgia mentioned that based on the Neoproterozoic oxygenation and glaciation events giving rise to the Ediacaran fauna, it was predicted that global environmental change during the Paleoproterozoic, about 2.4 billion years ago, may have also led to the development of more complex life. But until now, there has been no direct evidence of this theory due to a poor fossil record. Here we have two new forms, both displaying a previously unseen level of complexity at a time of significant environmental change. This provides strong evidence that biological complexity early on in Earth's history was also driven by environmental change. Our research shows that more complex forms of life were present earlier on in Earth's history than previously thought. So, bringing this to implications in terms of the search for life elsewhere, if relatively complex life in both macro and microscopic forms had developed this soon on the early Earth, then it is now seemingly more plausible that such a level of complexity could also have arisen elsewhere in the universe. Thank you very much.
Huge thanks to Erica and Georgia for that seamless and very enjoyable two-part talk. It's very educational, thank you. So I see that Georgia's been answering questions. So a question for Erica. So this is from Eva. So did you try some ramen on the organic matter of the fossils
Yes,
or other
question.
characterization?
Yeah, good question. We had such limited time in this talk, I couldn't go into it, but yes, I've definitely done a lot of ramen spectroscopy on these fossils, and also in situ carbon isotope analysis. So both of those have without a doubt confirmed the biogenicity of the fossil. Going further in terms of characterizing the type of organic material, more along the lines of what Bonnie was talking about in the previous talk, that work is still under progress, and actually Bonnie, as part of her PhD, is going into a bit more of that too. So watch this space for potential molecular biomarker results.
Great, and there are more questions in the chat, so I might direct you both there. So thanks for that, that was fantastic. So now we have our last short talk before our invited speaker. We have Keyron, so If you'd like to start sharing your screen, then we can check that it all looks okay.
Of course. Let's see.
Yeah.
Hopefully you can see this.
Oh, yeah, we can see it. So please check presenter mode.
I'll go-
Okay, this looks great, full screen.
Very much
Yeah.
so. Yes.
So pleased to welcome Keyron Hickman-Lewis, joining us from Europe. It's super early in the morning. Are you in Europe right now or in South Africa?
I'm in Europe at the moment and it's 5:45 a.m.
Okay, well, thank you so much. Please, the stage is yours.
Okay, thank you. So as I say, it's 5:45 in the morning in Europe, but what better way to start the day than with a healthy dose of deep time paleontology with all of you. So hopefully in this presentation, I will recapitulate and offer some perspectives on our research over the last 5 years, which concerns the reconstruction of the paleo-environments and ecosystems of the Palaeoarchaean. So the Palaeoarchaean is the time in Earth's history spanning approximately from 3.2 to 3.5 billion years ago, from which we have the oldest reliable and a well-constrained fossil record, and therefore the oldest archive of the co-evolution of the Earth and life. So in the talk, I want to explore 3 broad ideas. The first of these is that although we generally still consider the Archean to be a problematic world that's rife with uncertainty and suspicion in its interpretation, this really isn't and shouldn't be the case. That the Archean, although challenging, is a rich archive of paleontological and paleoenvironmental information. And a number of emerging and innovative high-resolution approaches are continually unveiling new details for us on Earth's early co-evolving geosphere and biosphere. Admittedly, there are very few localities from which one can study the earliest traces of life, although I would argue out of these, the Barberton Greenstone Belt of southern Africa preserves ancient fossils with perhaps the highest degree of individual fidelity. So most of our studies therefore focus on the Barberton Greenstone Belt and primarily on the widespread microbial mats, occasional microfossils, and rare stromatolites within. And most of these fossils come from volcanic clastic and black and white cherts, such as those in the photographs at the left of the screen. So the oldest traces of life in Barberton stem back to approximately 3.47 billion years ago. These are microbial mats in the middle marker horizon, which we described in a 2018 article in Precambrian Research. But as we show in the stratigraphic column on the right-hand side, microbial fossils and traces of life are found throughout the entirety of the stratigraphy of Barberton. So in this presentation, I'll make a specific focus on 4 examples of microbial mats, those being the 3.33 billion-year-old Josephsthal and Footbridge cherts, the 3.45 billion-year-old Huguenot Formation cherts, and the middle marker that I've already mentioned, and they are indeed represented in the photographs and photomicrographs at the left-hand side of the screen. So to understand ancient life, you need to understand its settings, and we studied the 4 horizons I've already mentioned with bulk and in situ laser ablation ICP-MS in order to measure the trace and rare earth element compositions that can tell us something of the chemistry of their depositional environments. So on the screen, I will show you some examples from the middle marker. So in almost all cases, bulk analyses, which we show here, are characterized by heavy rare earth element enrichment, positive lanthanum and yttrium anomalies that indicate marine precipitation. These are coupled with positive europium anomalies, which are diagnostic of hydrothermal influence. The picture changes, however, when we take high-resolution individual laser ablation points within microbial mats themselves. Here, by comparison to our original pattern shown here, the rare earth element pattern becomes flat, it becomes enriched in light rare earth elements, and it becomes devoid of any of the anomalies that I mentioned a few moments ago. Now, this is the telltale sign of terrigenous riverine chemistries. This indicates the presence of not insubstantial continental landmasses already at this time, and the trace element signatures from these land masses which enter into the microbial ecosystem. This is clearly sustained by a trace element systematics as shown in the top right of the screen. So clearly microbial habitats throughout the Paleoarchean were associated with complex aqueous chemistries which occur at the confluence of marine, hydrothermal, and terrigenous riverine environments, which are best interpreted as epicontinental basins with periodic marine recharge. So we can also use semi-quantitative mixing lines for the rare earth element characteristics, seawater, hydrothermal, and the Archean continental outflow to demonstrate this complexity. And moreover, as shown on the right-hand side of the screen, we conducted laser ablation ICP-MS transect analyses through microbial mats themselves at millimeter scale horizons. And these analyses invariably denote short periods of marine waters entering into the system, increased lanthanum, yttrium, and europium anomalies, and increased yttrium-holmium ratios are geochemically diagnostic of marine inputs into otherwise periodically restricted basins. So clearly, microbial ecosystems in the Paleoarchean are proliferating in periods of fluid disequilibrium, much as they do on the modern Earth. And the key take-home point from this part of the presentation is that understanding this complexity requires detailed in situ geochemistry in addition to bulk rock analyses which mask fine-scale fluctuations in geochemistry. So that's the environment, and now what of its inhabitants? So in our most recent study, which is published in Paleontology, I believe today, we used Fourier transform infrared microspectroscopy to identify relics of biomolecules and fragments of a biogeochemical complexity in the organic materials that form microbial mats. So although these mats have been metamorphosed to lower green schist facies, this does not preclude their containing traces of organic biomolecules. Paleoarchean microbial mats preserve relics of aromatic, aliphatic, carbonyl, and carboxyl groups that together may indicate precursor biology. So at the right of the screen, you can see some of the aliphatic complexity which is recorded in our samples, for example, methylene and N-methyl as recorded in the mats shown in the photomicrographs to the right. It's demonstrated from modern biology that the relative proportions of CH2 and CH3, that's methylene and N-methyl, in a bacterial fatty acid membrane ester lipids versus archaeal isoprenoid ether membrane lipids is among the fundamental compositional differences between these two domains of life. And the ratio of the CH3 to CH2 can therefore be used as a tool for biological identification, even in fossil material. Taking our 4 microbial horizons as an example, we can see that the Huguenot microbial mats are characterized by a low CH3-CH ratios, which are consistent with bacterial membranes. The middle marker horizons are dominated by high ratios, which are characteristic only of archaeal isoprenoids, while the footbridge chert and Josephsdal chert mats exhibit a wider range of medial values, which probably reflect a mixed consortium. And you can see on the right-hand side comparisons with both modern biology and these ratios in other examples of microfossils and carbonaceous materials in deep time. So we also performed a whole host of statistical treatments on these values, the most informative of which is the orthogonal principal components analysis on the left-hand side, which demonstrates that although the microbial mats comprise a diverse complement of organic materials, it is the CH3 to CH2 ratio, as shown in the plot at the top left, that defines the majority of variance in their biomolecular composition. And the key take-home point from this part is that this enables us, for the first time, to describe Palaeoarchaean microbial mats at a taxonomic level. We can say which of our samples were dominated by the domain bacteria, which were dominated by archaea, and which were probably dominated by a mixed community. In addition to biomolecular relics, Palaeoarchaean organic materials also contain a host of trace element signatures which can be interpreted within the framework of their original biomass. So we pioneered this idea several years ago, and you can read about it now in our recent Scientific Reports article from earlier this year. In brief, trace elements, particularly the transition metals, are well preserved within carbonaceous material of probable biological origin from the Palaeoarchaean, and if interpreted within a well-constrained paleoenvironmental context, this can reflect the metallome of the original biomass. Now, the metallome of biology reflects the chemical selection of elements according to the requirements of the cell. In other words, these are the inorganic parts of the metallocenters that make metabolic cellular nanomachines operate. And in Archaean carbonaceous materials, up to 3.5 billion years old, we see that numerous transition metals are strongly enriched relative to their levels within the matrix. And within individual recognizable microstructures, they are strongly enriched to similar levels. We can therefore make comparison with some of the metabolic requirements in extant biomass, and we can suggest, for example, that biomass in the fossil record that is particularly enriched in iron and nickel relative to the matrix may indicate methanogenesis, or that iron and vanadium denote some cycling of nitrogen by the precursor organisms. I hope that this kind of technique is the sort of geochemical approach that one can apply, for example, with the Mars 2020 rover, if indeed we find organic materials at Jezero Crater. This is the kind of technique that could be directly applied by the Pixel instrument. So I've shown you several examples of exceptional preservation of microbial signatures in terms of biogeochemistry in Palaeoarchaean time. And it's important to note that the agent of exceptional preservation in Palaeoarchaean biomass is a silica, and that, I would argue, is initiated within hours and completed within days, is a sensible explanation for the exceptional preservation of biogeochemistry that I've shown you so far. So I'd like to conclude with a brief statement of the fact that not only is rapid preservation ideal for archiving biogeochemistry, it's also ideal for preserving morphology with high fidelity in 3 dimensions. So in these kinds of high-resolution CT scans, 3-dimensional scans of microbial mats, as shown on the screen, we can identify certain aspects of microbial mat behavior. For example, undulating or tuft building in mat laminations, as shown in the yellow circle on the right-hand side, and the trapping and binding of sediment particles, for example, the red particle within the circle, as shown at the right. We've applied this technique throughout the geological record. Some of the samples we've used stem from the Dresser Formation, and despite the pervasive remineralization of the samples that we studied, we can nonetheless identify relationships between the macrostructure and the microstructure, for example, the building of cones and pinnacles, that indicate, by comparison with modern biological growth experiments, biological behavior. Unfortunately, I haven't got much more time to delve into that today. So that's the conclusion of the talk, and I'd like to return to the 3 initial points, which is that I hope this brief overview has convinced you that there is indeed much detail to be garnered from Palaeoarchaean microbial ecosystems. We require only to harness the techniques and innovation that are required to extricate it from its rocky prison. With time, we hope that an increasingly detailed picture of Archaean life should enable us to move away from studying ancient ecosystems on the basis of fossil by fossil, and that we can instead start considering the early biosphere in terms of interconnected biomes, interconnected metabolic networks, and indeed an entire biosphere, much as we know on the Earth today. So on that note, I'd like to conclude and extend my thanks to the organizers for giving us an opportunity to present in this very fine virtual conference hall. And I'd like to thank all of you very much for your attention. Thank you very much.
Thanks so much, Keyron. That's fantastic. Unfortunately, no time for questions, but I'll direct you to the chat. Bonnie has 2 questions, and I think some other comments in there from Erica as well. So stay awake for a bit longer and answer those.
I'll do my very best. Thank you.
Okay, thank you. Great. I might get you to stop sharing your slides as well. So. Yeah, so this brings us to our last invited speaker for this conference. And it's super exciting. It's closing this session on the topic that we started with, which is exploration and space. So our invited speaker today is Dr. Supachai Awiphan. Probably pronounced that incorrectly, but joining us from the National Astronomical Research Institute of Thailand. So Supachai is a researcher at the Institute, which I'll call NARIT for short. His interests include finding exoplanets and exomoons in transit and using microlensing techniques, which we heard about 2 days ago, and studying planetary atmospheres. At NARIT, he is an exoplanet and astrobiology science group coordinator. So without further ado, I'd like to invite our invited speaker today to tell us all about exoplanets and astrobiology in Thailand.
Yeah, okay. Can you hear me?
Yeah, I can.
Okay, and you can see my slide, right?
Yes, looks
okay.
Very good.
Okay, I think it should be good afternoon for you, but it's good morning in Thailand because now it's near lunchtime in Thailand. My name is Supachai Awiphan. I'm the science coordinator of the exoplanet and astrobiology in Thailand, and we are at the National Astronomical Research Institute of Thailand, or we can call NARIT. And today I will present the work on the exoplanet and astrobiology in Thailand. And first of all, I think I will divide my talk into 3 parts. The first part, I think I would like you to introduce you about what is NARIT, where is NARIT, and what we are doing in Thailand now for exoplanet and astrobiology. Now, for the NARIT here, we are very young still in Thailand. It's just 10 years. If you can see logo down here, it's just 10 years last year. That means we are very young still in Thailand, and research is still in the world. But we hope that we are one of the biggest astronomy research institute in Southeast Asia. And before that one, I think I will go to the all infrastructure in here, and one of the major one that is we are astronomical research institute. We have a lot of telescopes around the world. Yeah, the main one, I think I would go one by one a bit. The main one is in Thailand. That is a 2.4 meter. That is the largest telescope, optical telescope in Southeast Asia now, and I hope that in next few years, Indonesia, they will have around 3 meter class telescope. It is one of the biggest telescope in Southeast Asia today, and it's in Thailand. And now we will have, for next year, we will have the another big infrastructure here that is in Thailand, in Chiang Mai also. That is the 14 meter dish radio telescope. This one maybe can be used for the search for life in the future, for the something like search for exoplanet and search for the SETI research also. I think I will talk better in this presentation. It will be finished next year. And also we have a bunch of small telescopes around the world. One in Chile, one in China, one in US, and another one in Australia, I think. That is the robotic one. That means we can control the telescope in Thailand to observe the sky in all the part of the world. And the next one is our main infrastructure here. We are in Chiang Mai. That is in the northern part of Thailand. We have the called the Princess Swinson Astropark. You can see that we have, this is the main building that I sit now, and we have the planetarium for students, we have the observatory for the public, and it's the inside the planetarium we have the astronomy exhibition, and that means all the people can come to Chiang Mai to our astropark and learn about astronomy. And that means we not do just only the research, but we also do the outreach also. And we also have the laboratory lab. That means it have the optical photonic radio frequency satellite payload lab that is in our astropark here. Is the one of optical lab here where that we try to build some instrument like spectrograph, coronagraph to detect exoplanets. And also not only in Chiang Mai is here in the northern part of Thailand. We also have the observatory around Thailand that is for the public, that we want to give the knowledge about astronomy, and in the future it should be astrobiology also to people in Thailand. We have now in the Nakhon Ratchasima, that in the middle part, Chachoengsao, that is very near to Bangkok in the southern part of Thailand in Songkhla. And this is the one in Nakhon Ratchasima and Chachoengsao. It looks very nice that we can get the people to learn about the astronomy here. It's the Stonehenge, the mini Stonehenge here. You can see this. All of the observatory is have planetarium for public also. There is one in Songkhla, this just opened last year. You can see the view of the Sea of Thailand here. And the next step, in next few years, we will have, now we have the Thai Space Consortium. That is the next few years, we will try to send the satellite that built by Thai people, go to the space. That the first one will be just the remote sensing, that to look for the weather, remote sensing, everything. But in the next around 10 years, we aim that we try to build some probe to moon, or in the far future, like 20 or 30 years, maybe probe to Mars. I don't know, but we just start. But that is a good start for the little country like Thailand now, where we have the Thai Space Consortium. We will send the first satellite in next, I think it should be 2 or 3 years. And also not only that one, you can see that there is we are at NARIT, we are the International Training Center in Astronomy under UNESCO. That is held start from last few years. This is in operation in last year. And the thing is, this one, that means we are the training center in astronomy. That is the only training center in astronomy in the world. That means all the people around the world we will set up the training, and people from around the world, we can come to Thailand and learn about astronomy. And the thing that in last year, we held we call the NARIT School for Exoplanet and Astrobiology, or NARIT C. And we invite 3 people, and 2 is come from one is come from the Australia, that is Rob Wittemeyer, that is for exoplanet, and Professor Catherine Campbell from University of Auckland for astrobiology. And we hope to have this kind of school to give the training for the student every 2 years. I hope that next year we can hold the School of Exoplanet and Astrobiology in Thailand again, and invite some of the experts come to NARIT and teach the students, not only for the Thai student, but for Southeast Asia students. And also if in the future we try to have online, that means it can be the people from Latin America, Africa, to learn about exoplanet and astrobiology also. That is the future plan for the NARIT also. And also in the early part, I think I will talk about the 40-meter dish, and it's not finished yet, and we hope to finish the construction of the 40-meter dish in next year. But in the future, we think that this one can be used for the SETI, for the SETI research also. Sorry that is the banner is in Thai, but you can see that we have the talk from the Andrew Simmons, that is the director of SETI, and Professor Mike Garrett from JBCA from United Kingdom. That we try to help the SETI research in the future also. That is we can use the 40-meter dish to help to search for the SETI, the signal from radio signal outside the Earth. And there is a start that we give the public talk from Andrew and Margaret in Bangkok and in Chiang Mai here. That means now you can see that NARIC is very new, but we aim to have build the people to give the knowledge to people in Thailand and also join the world class research in everything. It includes the astrobiology also. Okay, I will talk about a bit about the research in NARIC here. In NARIC, we have the 5 key scientific research area here. It comes from the space weather, Earth climate, that is about understanding physics of the world. The third one is the exoplanet and astrology. And the fourth one is the cosmology. And the last one is optics. I'm the group coordinator of this group, the exoplanet and astrobiology. And in our group, it's very new. You can see that we have only 3 people here. That is me, we have Dr. Sinrat and Dr. David Martician. Both of them is work on the exoplanet for the radiology. We don't have any astrobiologists in Thailand now, but I hope that in the near future, we will have the first astrobiologist come back from University of Auckland. I hope that she will come back to Thailand in the near future. Now we have just 3 members. We have the cooperate with the 3 universities in Thailand, Chiang Mai, Mahidol, and Suranari. But even we were a small group, but now we can have more than 10 students that work in our group here. That is very good start from this group in Thailand. And the group is, it can be divided into 2 parts, exoplanet and astrobiology. I think I will go first for exoplanet, that is my expert. To detect exoplanet in NARIC, we use many techniques. The first one is the simple one, that is the radial velocity. That means now we search for the giant bright sky in the sky to find the exoplanet, and now we found around 9 candidates that found with our collaboration. 9 planets have been found. And also in the near future, we will have the spectrograph that built in Thailand. We call ExoSpec, that is the high resolution spectrograph to detect exoplanet. And we hope to have the resolution around 100,000, and the precision is around 3 meters per second. And this is the sum of the on-sky test last year. That is from the Betelgeuse spectrum, and this is the spectrum of the Betelgeuse. This is the calibration thorium argon. And we hope that it will be finished soon in the next year or next 2 years. And the next one is for the transit technique. We cooperate with the KEL team. That is the team to search for exoplanet. And we also the part of use our telescope in Thailand here. You can see that it's a pinpoint for the KEL team in Thailand and our telescope in China to find a new exoplanet here. And we found 1 exoplanet. It's called KEL22b. And not only this one for the microsync technique, I think we also cooperate with the Gaia follow-up team. We are the part that use our telescope to find to help to model the one of exoplanet here. That is the Kojima 1b, one of the brightest exoplanet that have been discovered. And also we try to model the exoplanet microsync signal using the tool called Marble. That is the one that is, if you look on the sky where you want to find the microsync signal, that is the one that we can use to find what is the area that we can use to find the microsync signal. And also I'm the part of the group of the Roman Euclid observation. I think you know about the Roman satellite that is the next talk on Wednesday, or extend on Wednesday already. That is the one of the biggest NASA mission in the near future for observation. And Euclid is the one of another mission for the ESA from European team to send to the spacecraft. If we join these 2, and what is we should find for the planet, that is this is our work with our colleague in Europe and US also. And also we try to build the chronograph to get the direct image of the exoplanet here. We call the new technique called evaluation wave chronograph that can be used for to detect the direct image of exoplanet in the near future. And I also work on exoplanet atmosphere also. If you know that when the planet is passed in front of the star, some of the light can pass through the atmosphere. Some of the light is scattered in the atmosphere of exoplanet. Some of the light is blocked by the cloud on the exoplanet. That means we can use this one to find out what is the chemical composition of exoplanet atmosphere. And now we try to have many things, like we try to have the ranking. We develop the ranking. What should we detect? Which planet that we should look for exoplanet? We want to optimize using the machine learning thing, that how it will be quickly to model exoplanet atmosphere. And also we do the observation with our telescope also. This is one of our result, that we detect that the blue area here, the blue wavelength of exoplanet atmosphere, is have the larger size of planet. That means the planet is have blue sky like the Earth, and that because the light pass to the atmosphere of planet and it's scattered like the haze in the Earth. That means if you go to this planet, it will be something like you can see the blue sky of this planet, and it is one of our results from the exoplanet atmosphere. And also we try to do the exomoon also. You know that now we detect around 4,000 exoplanets, but we have just 1 or 2 candidates of exomoon. Why we cannot find it? Because the moon is very small. And you can see that if you have the transit of the moon, it will have the very shallow depth here. If this one is 1% of the transit of the exoplanet, the moon is less than 1%. That means it's very hard to detect moon. Then we have the mode to model, get the new technique like to find the moon, because we think that if we want to have the habitable planet, you should have the moon. Because if you don't have the moon, it will be don't have the tide on the exoplanet. Even it have water, it will not have the tide, and that means the life maybe not form because it is not have the evolution of that planet. That means it is the thing that we want to find the moon outside the solar system also, and we try to develop the new technique to detect it. And after I talk about the narrative for a bit now, I think for this talk, I will go to 2 of our projects that is in narrative here that we do closely related to astrobiology. The first one is the project called the Sky Area to search for extraterrestrial intelligence. And this one, I think you know that, okay, now we want to find alien or ET, but if we can detect it and we cannot communicate with them, that means because they cannot detect the Earth also. The idea of this project is, if you are alien, where is the area that alien can detect the Earth as the planet in the solar system also. That is the idea of this project, that we can detect them and they can detect us. In this project, I assume that the alien or ET have the same technology as the Earth at the present day. That means we know that we know few techniques that can be detect exoplanet, and if they use the same technique to detect the Earth, where to find them. And the first one is the transit technique. If you are the observer outside the solar system, you can see the Earth transit in front of the sun, but the angle that it can be detected is very small. We call it is the Earth transit zone, and it is the area here is just about 0.5 degree here. Angle here is just 0.5 degree. And if you plot on the sky here, it will be just a small red line here that is on the ecliptic plane that we the star in this slide can detect the Earth as the transit, because the other in the other area they cannot detect the Earth transit because the Earth cannot pass through in front of the sun. And the blue dot here is the confirmed exoplanet nowadays, and you can see that they are very narrow area here that we can detect the Earth is the galactic center here, and I think LMC and SMC should be around here. And for the radio velocity technique, it's quite the same. I think it's because will be better than the transit, because you can detect even the inclination, the angle is more than 90 degrees. It cannot be passed through, but the area is I think it should be something like large, like this one. We can set up the detectability to be 1. That is the maximum one that is near the ecliptic plane here, and this for the radio velocity technique. But as the Nick told present on Monday, on Wednesday, and I think you have seen this part already from Nick, that from the Kepler mission, I think if we want to detect Earth, it's near the edge of the detection limit of the Kepler mission. But for the Roman telescope or the Widefield telescope, we have the chance to detect the Earth more than the Kepler mission. And that is why, if we look for the microlensing technique, where we should find the observer that can detect the Earth via the microlensing technique, that is another question. And the thing is, now, if we have something like the alien here, that to look to the Earth and have the sun here, Earth and sun here, and should have the background source here. That means you can see that now the Earth acts as the lens here, and we have the one component called the Einstein radius, that is you can calculate from the mass of the lens and the distance between source and lens. Source and lens here, that is ds, and the distance between a source and observer, and the distance between lens and observer here, dl here, and we call this one is Einstein radius. That is the radius that we hope to detect the strong signal of microlensing. And if we want to have, that we have for one star here, you have a lot of the background star. That means we want to compute the microlensing occurrence probability. We call this one optical depth. That means we sum all of the Einstein area here, Einstein radius here, and divide by the total area of observation. As I told you, that is now the observer, we have the solar system as the lens, and we have the source to be antipole, that is the other side. And this we simulate based on the Gaia DR2, that is the largest catalog that we can have now. And with we add the extension from the Gaia extension also, and we limit that the star that can take the Earth here should have the magnitude brighter than 17, and they also have to look for the source that have the magnitude better than 17 also. And the result that show that this is the total optical depth here, and the area that have the highest probability is near the galactic center and antipole of the galactic center here. That means these two areas is the area that have high probability that the alien can detect the Earth. And that means if we want to find alien, that means both of these areas can detect the Earth easily. That means we should look for these two areas to search for something like the extraterrestrial life, because they can detect the Earth. That means if they can detect the Earth, maybe they have sent the communication to the Earth already. And it's the other one, if we look for the average, it seems like it should be quite the same. And that means in conclusion for this work, if we assume that the extraterrestrial life have the same technology advanced as the current human, the galactic bound and galactic anti-center are the two areas that we should search for extraterrestrial life. But I think, as you know, that this work is ongoing. We try to limit something like the galactic habitable zone to look for what is the habitable zone for the, because if you go to near the galactic bound, it will be not very good for the life to host, because you have a lot of supernova, many things. And for the second one, the second project that I would present, that is related to astrobiology, but I would say that it's very early project, and it's a pilot project to search for astrobiology in Thailand. I would thank Dr. Siramat and Chenoa, that is the PhD student in Auckland that create this work, and that's one of the guy that want to tell you. I don't know about it very in detail of this project a lot, but this project we want to present the characterization of Sinter from the Pūnaha hot spring, then analog to early Earth and the Mars environment. That is the one that we search for look for the hot spring in Thailand, and we found that we have one of hot spring that near our headquarters here in Chiang Mai. It's just 81 kilometers outside of Chiang Mai. It's called Pūnaha hot spring. It's located in Pūnaha National Park, and it has a geyser that in the middle of forest area. The highest geyser is about 1 meter high, and we hope that the micro species that we can detect here should reveal the diversity and ecology relation of this hot spring, and we hope that they can apply to paleogenesis and the evolution as extreme morphologic species at the root of the tree of the life in here. And this project is started in, I think, 3 years ago at the Pūnaha. We collect the sample. It's the hot spring, and we go deeper, and we have to have the permit from the national park to collect the sample. This one is not for the tour. I think it's open for tourists, but very few is known that it have this hot spring in Thailand. That means it have been preserved, not have for the tourist site. In one of the have been preserved to be the good site to collect some sample, and it's when we collect a sample here, and it's some sample that we get, and from the physiochemical property that we collect from the water chemistry, and we know that we collect two things: is near the waterway and near the geyser, and the temperature here is around 71 and 90. The pH is around 9, and we know that the chemical composition that we get is around mostly is the sodium, chloride, potassium, and we try to conclude that this Pūnaha is the alkali chloride hot spring. And after that, we do the simplification. That means we use the same image below here to see the zoom in, and we have showed the image. It shows that it have the different level of microbial simplification in the Sinter, and all sample point all sample from all collection point is showing quite the same level of the pattern of simplification, and also they have the large shape, the filament concord, like you can see here. And for the mineralogy, we use the XRD in Chiang Mai. We are very early, and we found that the chemical composition, the mineral found in the Sinter is have the effect of the XRD around 22 to 22.2 to 22.4. That is confirmed that the Aphomerus silica and other mineral that is found in the Sinter is like quartz, microlite, and calcite, and some of the ammonium. And this is the mineralogy that we found from the sample that we get, and we also do the RNA to find the taxonomy diversity and species distribution. And most of the species that we found is the prokaryote of the chlorophylls and cyanobacteria and the proteobacteria. And this is the result from the RNA technique. And in the conclusion, that is now we have the mineral result that obtained. That is the primary result from the present. The sample is show that we have the orthogonal silica in the deposit that is quite near found in the Barlow spirit Sinter near the Colombian Hill. And we found that several bacteria that is report in this work to have the different temperature and the pH condition than the previous literature. And this is just the start of the astrobiology work in Thailand. I know that after I come to this conference, I know that you guys go beyond us a lot, but we just start, and we hope that in the future we can collaborate with your guys to help to set up the study of astrobiology in Thailand. And that is all for my talk, and thank you.